Hi everyone, I'm Elena, and today I'm going to be presenting on my Keystone as done through the Honors in Biochemistry and Molecular Biology pathway in Dr. Mikel Garcia Marcos' lab, entitled A High-Throughput Screening and Identification of G-Protein Binding Peptides. So to start, G-protein coupled receptors, or GPCRs, are the largest class of transmembrane receptors. When a ligand or a small molecule binds the outside, that leads to a conformational change in the GPCR that activates the associated G-protein. So why are they important? GPCRs are physiologically relevant due to being heavily implicated in signaling a lot of our senses as well as higher mental function. That means that when they're dysregulated or mutated, that could lead to many different diseases as seen on the left. Because of this, they're also strong pharmacological targets. 30 to 40% of FDA-approved drugs actually target GPCRs. And over here, you can see some areas of interest for GPCR targeting, including diabetes, Alzheimer's, and obesity. In addition, GPCRs primarily signal through heterotrimeric G proteins with an alpha, beta, and gamma subunit. When the GPCR is activated, that leads to the activation of the G protein, and that key moment of activation is when the GDP on the G alpha, marking the inactive state, is displaced to a GTP, marking the active state, sort of like a light switch. In addition, there's also 4 major families that initiate specific signaling pathways. So, because they're a complex way of activation as well as physiological importance, there's a need for tools that can measure G-protein activity in a proximal and specific manner. One way to do this is through detecting G-alpha GTP itself. So, why would we want to use these tools? For one thing, for drug development, to understand any unintended side effects of drugs that target GPCRs, as well as understanding the specific mechanism of activation for GPCRs we know less about. So in the past, my lab has done this through creating different biosensor platforms. They have many different components, but today I'm just going to focus on one key component, boxed in red, which is a peptide or small protein that acts to bind the active state, acting as a tag. These peptides are ideal for biosensor usage because they're specific, they're high in affinity, and they're also reversible in binding to G-alpha GTP. We found these peptides from a patent. However, this patent's been largely unexplored for its potential. Therefore, here lies the goal of my project, which is to conduct a screen of 116 peptides from this patent to identify ones that preferentially bind the active or inactive state of G-alpha subunits of the 4 different families. We want to do this to optimize the biosensors we already have, to find potential inhibitors of G-protein activity, as well as to further understand the structural functional relationship between this protein and peptide binding. Why do they actually bind together? With this many conditions, our approach for this project is going to be doing a high-throughput screen using bioluminescence resonance energy transfer, or BRET, in HEC293 T cells. So to go into my methodology, bioluminescence resonance energy transfer, or BRET, is an assay involving light with 2 key components. You have a BRET donor, I'll be calling the N-Luke, and a BRET acceptor, which is our YFP. When these 2 signals get really close together, we get a high BRET ratio. So if we take this a step further, we fuse one of the N-Lukes to the peptide we're testing, and we fuse a YFP to the G protein we're testing. If the peptide and protein bind each other, we get a high BRET ratio. We can take this another step further by, for each tested N-Luke peptide, we can transfect it with 2 different conditions. One being a wild-type inactive G-alpha GDP, and the other being a constitutively active G-alpha GTP. We can then take endpoint measurements, and whichever shows a higher BRET ratio indicates higher affinity binding for that particular G protein state. So if we test a certain peptide and we see that the inactive state shows a higher BRET ratio, that indicates that peptide preferentially binds the inactive state over the active state. We can then perform this for each G protein family. So we did this screen and we got a lot of data using a set of controls as well as each peptide we were testing. So today I'm just going to focus on one particular G protein, G-alpha OA, which is a member of the G-alpha IO family. So we first started with a set of controls, and for our control of binding to the active state of G-alpha O, we have KB1753, and for our control for binding for the inactive state, we have R6A. We then, from our screen, found 7 different peptides that preferentially bound to the active state, and what was interesting is that they actually bound with a higher affinity to the active state compared to our previous control of KB1753. So why might this be important? Well, G-alpha OA is really physiologically relevant, it's highly active in the nervous system, it's overexpressed in certain cancers, such as certain breast carcinomas, as well as being implicated in a neurodevelopmental disease called JNL1 encephalopathy. So our next question is, we found these peptides, but we want to see if we can actually use them in a useful way, for example, through biosensor usage. Because in theory, if they bind really highly to the active state, even higher to the standard, we'd expect they can maybe be even better biosensors. So our next question is, if these peptides can be applicable as detectors for GPCR-mediated activation. So we decided to assemble our newly identified peptides into one-vector plasmid biosensors. We selected 3 of the peptides from the last slide, and we decided to compare them to KB1753, which is our usual control for J-alpha-O activation within our biosensors. So to actually test if these biosensors work, we decided we were going to do kinetic activation by testing a GPCR that couples through J-alpha-O, and then when we add an activating molecule, do we actually see that increase in breadth that indicates activation? So we decided to use the dopamine 2 receptor, which couples through J-alpha-O, and what we expect is that when we add our agonist of dopamine, we get the activation of the GPCR that leads to the activation of J-alpha-O. And if these peptides are working in this biosensor, we expect to see an increase in change in breadth upon this occurring. What we find is that we were able to leverage and use these peptides to detect J-alpha-O activation upon GPCR-mediated stimulation. As we can see, upon dopamine addition, we get an increase in delta breadth. Finally, as I learned the hard way, screens can take a really long time. So ideally, what we'd like to be able to do is bioengineer a peptide that binds exactly how we'd like to a particular G protein and state. However, to do this, we have to understand more about why are these G proteins and peptides binding each other. What building blocks within the peptide, otherwise known as amino acids, are important for this interaction to occur? So we decided to do a proof of concept experiment using a case study of J-alpha-S and KB1691 to test this out. As we can see here, KB1691 preferentially binds the active state. So we decided to do this doing site-directed mutagenesis by changing each individual amino acid within the peptide and combining it with the active state of the G protein to see how does this change the signal. If we get a loss of signal upon mutagenesis, that would indicate that that amino acid was key for that interaction. We can then also compare it to a computer model called CollabFold that can predict how this interaction is going to occur. What we find is that these 5 amino acids were essential for the KB1691 and J-alpha-S GTP interaction. And when we looked, we see this because the signal decreases upon mutating that amino acid. We then looked at the computer model, and we found that it actually aligned super well to these results, so we now have a way that we can actually test our protein-peptide interactions. For our conclusions, we were able to successfully scale up and develop a high-throughput screening assay for 116 peptides. We identified novel peptides with the potential and applicability to utilize as biosensors, inhibitors, and modulators. And we developed a proof-of-concept experiment using site-directed mutagenesis that we can use to examine protein-peptide interactions. For future work, we can look at testing more families and applying more of these peptides in useful ways, as well as also doing the site-directed mutagenesis of the hits we identified to improve the potential bioengineering and design of a peptide that binds exactly how we'd like. Overall, we now have more to use in our toolbox of sensing, inhibiting, and modulating GPCR and G protein signaling, which has large potential for drug discovery and monitoring disease progression. For my acknowledgements, I'd like to thank the entirety of the MGM lab, with a special thanks to my PI, Dr. Garcia Marcos, as well as my thesis committee, which I recently defended on Wednesday and passed, as well as my support and funding sources over the years. Thank you.
What drove you to do this research, I guess?
I've been in the lab since freshman year in this particular lab, and I think the drug potential with GPCRs is so vast, and there's so much that we can potentially do. Like I mentioned earlier, 30 to 40% of FDA-approved drugs literally target GPCRs. There's so much we can potentially do, and so this is one stepping stone to really get a broad look at the many different ways we can use peptides that bind G proteins in so many different ways, and yeah, I think it's a good step towards that, and that's definitely what drew me towards this specific project.
What was your favorite part of the project?
I think definitely this last part I did with the mutagenesis experiment. It was a pretty novel experiment for my lab in regards to actually exploring the structure of KB1691. And actually getting really meaningful results with the fact that it actually aligned with our computerized model. We did not expect that to occur at all, and it actually did, which was super exciting. Yeah, and it's also now it's now on the way to publication, which is also really exciting. Yeah.
What were some of the biggest obstacles you faced doing this entire project? Because I'm sure it was
Yeah,
probably...
actually developing the screen itself took more than a year. It took like actually, because we started with a lower throughput setup of 6 wells, and then we moved on to a 96 well plate setup. Making that transition took a really, really long time, just adjusting different concentrations, all of that. It took so many different tries. The graduate student I work with closely, Remy, we spent so many hours looking over the data, trying to figure out the right way to do it. And eventually we got it to work, but that was definitely the hardest part, was actually getting the screen to happen.
Could you actually talk a little bit more about that screening process? Because it sounds incredibly laborious. But what do you like? Can you just tell us a little bit more about what you're physically doing to make that happen?
So we do this in a 96 well plate setup, and we split up a bunch of cells basically into each of the wells. And for each peptide we're testing it with 2 different conditions: the inactive state and the active state. So we seed the cells, we put the cells into the plate, we then add in the components of these KB peptides and the G proteins in a method called a transfection. And then the next day we actually do the actual experiment of putting in the Brett components and actually seeing what signal do we get. So it's sort of like a 3-day cycle in a sense.
All right, thank you so much, Elena. Please come back at 11:45 for Cameron's presentation.